So similar to prokaryotes, eukaryotes also have to regulate their gene expression. But it's going to be a little more complicated, okay? So prokaryotes, the big thing is operons and usually 2 levels to control them. Here we have a lot of different factors to consider. So for note, under normal circumstances, all cells contain the same DNA. Okay, so this is barring any mutations. So why do cell types look different? Why does a liver cell resemble this orientation, whereas muscle cells are longer, shared nuclei, etc.? Okay, so the answer is going to be largely or even loosely gene regulation. And to that end, why else might a eukaryotic cell need gene regulation? And the answer still should be similar to what you would say if I asked you about a prokaryote. Respond to their environment, all right? So think about what happens if you go outside in the sun. You usually tan. This is your body's way of responding to your environment. Or think about what happens to a sunflower. They turn towards the light. They are responding to their environment. If there's a change in nutrient availability. So when we talk about eukaryotes, remember, we're not just talking about animals or we're not just talking about plants. There's still protists and fungi out there. And so they need to respond to the nutrients available to them for development. As we go from an embryo to an older person, we are changing several times, and we have to be able to produce the proteins we need to accommodate those changes and then turn them off when we no longer need them. And then, as stated in the previous slide, to be able to create different tissues and cell types. Okay. Now, I want you to go back to last module and write down non-coding RNAs that play a role in gene regulation. Okay. So, again, similar. You should see like large concepts remain the same, that this gene expression can be controlled at many different stages, from the DNA to an active protein. And which step is the most important? And your answer should be the same, transcription. Transcriptional control is paramount because this allows you to not even make a gene. So you're not going to waste energy going through all these different stages just to not use that protein, okay? And so there's all these different factors that can be controlled to control transcription. So to reiterate, we can control gene expression at the transcriptional level. Remember that post-transcription or co-transcriptional / the RNA processing. We can control it translation or even post-translational modification. So we have several levels that we can intercept. All right, so when we look at the transcription, remember those transcription factors? Remember that T, F, 2, D, and all of those things? So these are places where we can control. Remember that transcription factors, their job is these basal levels. So they are there largely to help us get polymerase and do what we have to do. In addition to those, if we want to affect the rate of transcription, we have regulatory transcription factors that bind to regulatory elements. So in eukaryotes, we have an activator that speeds up, and an activator's job is to bind to an enhancer. So here you have an activator, we have an enhancer upstream of our core promoter. Or you have a repressor who binds to a silencer. So we have a silencer upstream of the promoter, and the repressor binds to it. Oftentimes, though, we have this combinatory control where it's not just one thing that's controlling it. And again, think back to the lac operon. They had 2 levels of control. So here we're talking about it's not always just one way we control. So we can control it up to 5 ways. We can use all 5. We can use a couple of these 5. So the first is requiring one or more activator. Okay, so we have to have one or more activator present in order for polymerase to work. Or we have to have one or more repressor present in order to stop polymerase from working. Remember those effector molecules. So we can require a effector molecule or 2 proteins to interact or covalent modifications like phosphorylation in order for something to happen. Remember those nucleosomes. Remember euchromatin versus heterochromatin. So we can control the arrangements of nucleosomes to affect transcription. And we can also methylate. So think back when we talked about epigenetics and DNA methylation, okay? So these are all ways that we can affect transcription, and we're going to go into detail about some of them. So in general, the role of these transcription factors is going to be to upregulate or downregulate transcription, or turn genes on and off, as we commonly say. For upregulation, we can upregulate 10 to 100-fold by having these activators. These regulatory molecules tend to have orientation independent, so they can go either way. Okay, so here you see GATA, ATAG, or GATA backwards. So they can be independent of what the orientation is, and they can be located at a distance, so they can be near the gene or further away from the gene. And this is possible by them just causing our DNA to actually change the way it's shaped, like physical changing. So it's possible to loop the DNA so that the activator interacts with its binding site. Okay, so let's look at the roles of these activators and repressors. So we want to be able to stop the initiation of transcription. And so one of the main things would be TF2D. Do you remember TF2D? So TF2D had the TBP binding site. So TF2D had our TATA binding protein as part of it, which binds to the TATA box, which was the first step of eukaryotic transcription initiation. So if we want to control transcription, we can control it at the level of TF2D. So how do we do that? We can do that by requiring an activator and a co-activator to come on and bind to the enhancer to allow TF2D to work. Or we can have a repressor that binds to a silencer and physically stops TF2D from binding. Okay, so if TF2D can't bind, it can't recruit polymerase, we can't have transcription. Or if we help TF2D bind, then maybe we're going to speed up the rate at which we transcribe. Remember mediator. So mediator's job was to switch between initiation and elongation. So we can have an enhancer that comes in and helps recruit mediator so that it can function for that switch from initiation to elongation. Or we can have a repressor comes in that stops mediator from doing its job, and then we can't switch from one to another, okay? And then we can't have transcription. So this begs the question, why is it necessary to modulate these transcription factors? Well, think back again to the beginning when we said, why is it necessary for gene regulation? We have to be able to control when these genes are on. We need them when we need them. We need them where we need them. And we need them under the conditions how we need them. Okay? So we have to be able to control these things. And the easiest way is to control what controls the gene, so those transcription factors. Now, we can modulate these three ways that we talked about. So remember those effector molecules. So if we have an effector molecule that binds to our transcription factor and it changes the shape, thereby allowing that transcription factor to bind to the DNA. We can have transcription. So there's these protein interactions where they have to be together to bind to the DNA. Any single one can't bind to DNA alone. They have to form a dimer, and together they bind to our DNA. Or we can require modifications like phosphorylation that's necessary in order for this to be recognized or to start transcription. So these are three ways that we can control our transcription factors. Okay. So now let us consider an example, real life example in eukaryotic cells. So we're going to look at steroid hormones. So remember, hormones are secreted by the endocrine glands. They have to travel through the bloodstream and then enter the correct cells, okay? So think about that. Now, we have these glucocorticoid steroids. So they are released into the bloodstream, okay? They're able to enter through diffusion because they can get through the plasma membrane. So they diffuse inside of the cell. Once they're there, they bind to their receptor. Okay, so the glucocorticoid steroid comes in, binds to the correct receptor. This receptor is turned, let's think of it as being turned off in a way. So it has these heat shock proteins that are binding to it. Once the steroid binds to it, it releases those heat shock proteins. When it releases those heat shock proteins, it now shows a nuclear localization sequence. So remember those SRPs. So now it has a sequence that will allow it to come inside of the nucleus, okay? And so we bring it inside of our nucleus, and when it's in there, so it forms this homodimer, comes inside the nucleus. So now remember, these are different steps. So here it has to work as a homodimer. That homodimer then goes and binds to this GRE sequence, and now we're able to transcribe. So this is multiple levels. First, it has to get inside the plasma membrane. Then it has to find its receptor. Its receptor undergoes a conformational change that exposes a sequence and allows it to form the homodimer. So then it travels into the nucleus, binds to the DNA, and helps with transcription. Okay? So any questions, comments, concerns? Let's talk about it.